Hello friends, today in this video we are going to start a new series of videos on MCQs on biochemistry. Okay, and this is the part 1 of the series of videos on biochemistry. So let's start. The first question is, question number 1 is, which is true? And options are: option A, holoenzyme equals to apoenzyme plus endoenzyme; option B, apoenzyme equals to holoenzyme plus exoenzyme; or option C, coenzyme equals to holoenzyme plus apoenzyme; or option D, holoenzyme equals to coenzyme plus apoenzyme. And the right answer is option D, that is holoenzyme equals to coenzyme plus apoenzyme. Okay, so let's move on to the next question, that is question number 2. So the protein part of an enzyme is option A, prosthetic group; option B, apoenzyme; option C, holoenzyme; option D, zymogen. And the right answer is option B, that is apoenzyme. So the protein part of enzyme is apoenzyme. Okay, so next question देखेंगे. Next question है, the substance which inactivates an enzyme by denaturing is called option A, enzyme inhibitor; option B, allosteric inhibitor; option C, competitive inhibitor; or option D, irreversible inhibitor. So the right answer is option D, that is irreversible inhibition. So the substance which inactivates an enzyme by denaturing is called irreversible inhibitor. Okay, so next question है, the structural unit of living system is referred option A, DNA; option B, RNA; option C, cell; option D, factor. And the right answer is option C, that is cell. So the structural unit of living system is referred cell. So let's move on to the next question, that is question number 5. The enzyme which combines with non-protein part to form a functional enzyme is option A, holoenzyme; option B, prosthetic group; option C, apoenzyme; or option D, none of these. And the right answer is option A, that is holoenzyme. So the enzyme which combines with non-protein part to form a functional enzyme is holoenzyme. So next question है, question number 6. So the science that deals living thing and their chemical processes is referred to as option A, molecular biology; option B, biochemistry; option C, bioenergetics; option D, bioanalysis. And the right answer is option B, that is biochemistry. So the science that deals living things and their chemical processes is referred to as biochemistry. Okay, so let's move on to the question number 7. So it is, in Ramachandran plot, the values of the dihedral angle psi is based on rotation around option A, N C alpha bond; or C alpha C bond; or C alpha or C N bond; or N and H bond. And the right answer is option B, that is C alpha C bond. So in Ramachandran plot, the values of the dihedral angle psi is based on rotation around C alpha C bond. Okay, so let's move on to the next question, that is, ribosome is RNA with extra phosphate, RNA without phosphate, RNA without sugar, or RNA with enzyme activity. And the right answer is option D, that is RNA with enzyme activity is known as the ribosome. So let's move on to the next one. So enzyme with different molecular configurations but with same function are called isoenzymes, apoenzymes, coenzymes, or inducible enzymes. And the right answer is option A, that is isoenzymes. So enzymes with different molecular configurations but with same function are called isoenzymes. Okay, so next question is question number 10. So restriction endonuclease is employed for joining strands of DNA, cutting RNA, cutting single-stranded DNA, or cutting double-strand DNA. And the right answer is option D, that is cutting double-stranded DNA. So restriction endonuclease is employed for cutting double-strand DNA. Okay, so next question देखेंगे. Next question is inactive zymogen, that is proenzyme, are precursor of all the following gastrointestinal enzymes except trypsin, chymotrypsin, pepsin, or ribonuclease. And the right one is option D, that is ribonuclease. So inactive enzymes, that is proenzyme, are precursor of all the following except ribonuclease. Okay, so next one is which one of the following statement correctly describe allosteric enzymes? And the options are they are not as usually controlled by feedback inhibition. The regulatory site may be the catalytic site, or Michaelis-Menten kinetics describe their activity, or effectors may enhance or inhibit substrate binding. So the right answer is about the allosteric enzyme is option D, that is effector may enhance or inhibit substrate binding. So the correct answer about the allosteric enzyme is effector may enhance or inhibit substrate binding. Okay, so move on to the next question. So which one of the following amino acids interrupts a helix and also disrupt beta sheet? And the options are PHE, that is phenylalanine, or cysteine, or histidine, or proline. And the right answer is option D, that is proline. So proline, which is an amino acid, interrupts a helices and also disrupt beta sheet. Okay, so let's move on to next question. Question number 14. So which among the following is or are methods of separating and purifying biomolecules? And the options are cell fractionation, chromatography and electrophoresis, or ultracentrifugation, or all of the above. And the right one is option D, that is all of the above options are correct about the methods of separating and purifying biomolecules. Okay, so next question देखेंगे. Next question है why does water is considered as universal choice of biological solvent? So options are water is dipole, or it is formed of hydrogen bonds, or interaction with water influence biological structures, or all of the above. And the right answer is option B, that is it is formed of hydrogen bonds. So that is the reason, that's why universal choice, that's why water is considered as universal choice of biological solvent. So next, move on to the next question, that is which among the following will be method of choice for determining biomolecular structure? And the options are mass spectroscopy and NMR analysis, use of protease, nuclease and glycosidase to degrade biomolecules for study, or sequencing of nucleic acid and proteins, or all of the above. And the right one is option D, that is all of the above are correct about the method of choice for determining biomolecular structures. Okay, so चलिए next question देखते हैं. Next question is regulation of fatty acid biosynthesis occurs at the enzymatic step catalyzed by carnitine acyltransferase 1, or acetyl-CoA carboxylase, or pyruvate carboxylase, or citrate synthase. And the right answer is option B, that is acetyl-CoA carboxylase. So regulation of fatty acid biosynthesis occurs at the enzymatic step catalyzed by acetyl-CoA carboxylase. Okay, so let's move on to the question number 18. That is the Ramachandran map of a protein representation allows you to identify the most stable structure, or the tertiary allowed structure, or the sterically disallowed conformation, or the secondary structure elements. And the right answer about the Ramachandran plot is option C, that is the sterically disallowed conformation. So the Ramachandran map of a protein representation allow you to identify the sterically disallowed conformations. Okay, so let's move on to the question number 19. So in which of the following cases the enzyme substrate complex is irreversible in nature? And the options are competitive inhibition, non-competitive inhibition, uncompetitive inhibition, or both competitive and non-competitive inhibition. And the right answer is option C, that is uncompetitive inhibition. So in uncompetitive inhibition, the enzyme substrate complex is irreversible in nature. Okay, so let's move on to the last question of this video, that is question number 20. So the term primary structure of protein refers to sequence of amino acids, subunit interaction, or overall conformation, or all of these. And the right answer is option A, that is sequence of amino acids. So the term primary structure of protein refers to sequence of amino acids. Okay, so these are all 20 questions on biochemistry. Okay, so thank you for watching this video.